let's get started at next session. I'm chair of this session, Akio Kanai, KOU. I'm really happy to introduce Dr. Deamer. He's a professor of the Department of Chemistry and Biochemistry, University of California, Santa Cruz. The chemical evolution and hypothesis to explain how life might be possibly evolved from the non-life. He's one of the expert of this particular field. Today, he's going to talk about his recent result entitled the self-assembly process in the prebiotic environment. Please welcome Dr. Deamer.
I want to join previous speakers in thanking Professor Rossi for the wonderful symposium he's put together. My compliments. I enjoyed it very much. So previous speakers have each given us a facet of the answer to the question of what is life, and each facet adds together, and I haven't seen the elephant yet, but I'm going to add my own facet now, because I'm also going to talk about what was life. I think part of understanding what is life today in the present tense is to understand where it came from in the past tense. And another facet yet is to think about what will life be, because the fact is that we're probably on the edge of putting together artificial forms of life. I'm going to bring you an up-to-date on that in the last portion of my talk. And I might add that quite a bit of the advanced work being done is being done in Japanese laboratories, including that of Professor Kaneko, who's spoken to us earlier. Well, understanding what is life begins with a definition, and I had Sergei Sokolov in my lab as a Fulbright scholar last year, and this is his poster right up here. He tried to put together his impression of why it is so hard to define life. And the problem, of course, is that life is very complex and requires a rather complex definition if we're really going to separate it from other things that resemble life. Now, to give you some idea of this complexity, what I'm going to try to do is to define a Toyota, nice local complex device. And first thing to do is to distinguish whether I'm talking about the city of Toyota, or Mr. Toyota, or the car. Well, that's easy to do. I can say that a Toyota is a four-wheeled vehicle used for personal transportation, and that leaves out Mr. Toyota. However, this also defines a cart, a pony cart pulled by a horse, so I've got to do a little bit better. A Toyota is a four-wheeled vehicle with an engine that is used for producing, for personal transportation. Well, unfortunately, that could also be a pony cart with an engine in the back, taking the engine off to the mechanic to be repaired. So I've got to do a little bit better than that. So let's try again. A Toyota is a motorized four-wheeled vehicle used for personal transportation in which the engine causes the wheels to rotate and produce forward motion. Well, I'm getting closer, definitely getting closer, but it needs fuel. I can't have a car without an external source of energy. So let's try adding some fuel. A Toyota is a motorized four-wheeled vehicle used for personal transportation in which the engine uses fuel to cause the wheels to rotate and produce forward motion. But of course it needs a steering wheel. It's a motorized vehicle with a steering wheel, etc., etc. And finally, I've come close to defining a Toyota, but I've also defined a Honda. So that's a little problem. I have to look at the history of the cars, because Toyotas originate in Toyota factories and Hondas originate in Honda factories, and the same in defining life. If we're going to learn what life is, I think we have to learn more about its history. Furthermore, with the car metaphor, we need to define the engine, the fuel, the steering wheel, so all the pieces of the definition also depend on what whatever we want to put together as a definition. So let's think now about life. It's complex, and therefore the answer must be complex. Those are the same sort of thought process very quickly. What is life? Well, all life is cellular. We agree to that. So the minimal unit of life is a cell. So if I can define a cell, I'm well on my way to defining at least the simplest kind of life. let alone the sort of lives that we live. Well, a cell is the unit of life. It's a microscopic compartment bounded by a lipid bilayer. The compartment contains polymers called proteins and nucleic acids. The proteins are catalysts, and the nucleic acids contain genetic information. Oh, is that alive? No, it's not. There's more to it. I can do this in my laboratory. In fact, we're trying to do this. We are building lipid bilayer vesicles, and we're putting enzymes and information into it. And we haven't come up with life yet, but we're trying. So let's see what's missing. And here is what's missing, all of that. Now, I claim that this defines life in the sense that nothing else in the universe fits this rather long and complicated definition. Furthermore, each of these words I've highlighted in green are keywords. And if I leave any of them out of this definition, suddenly we are less than something that we would call alive. So we're pretty close to a working definition of a life if we define the simplest life as a single cell. Well, like Toyota, we do still have to define the key words. So if I really wanted to define life, I would teach a course in cell biology. And I would start to define all of these words chapter by chapter for you. And finally, we would get up to something that we would call alive. Well, we want to go beyond simple description of life. We want to understand at a deeper level. So let's ask, how did it begin on the early Earth? And let's also ask whether we can synthesize life. Do we know enough now to put together a living cell, either by reassembling the pieces from previously living cells, or by actually starting from scratch? And as I said, we are coming close. Well, life is not just on the Earth now. We have a different definition. We're going both temporally, both forward and backward in time, but we also want to go out into the space around us. And there's a new field of research that I'm involved in. It's called astrobiology, in which life is defined as being part of a universal process. So the best way to think about the origin of life is to go back to the beginning of life in the form of biogenic elements. There's 6 of them, and there they are. Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. If we took a living cell out of one of our bodies and analyzed it, we would discover, putting aside things like potassium and chloride and so forth, we would discover that these biogenic elements and the molecules that they compose are something like 99% of the mass of a living cell. So these are the important elements. We can say, where do they come from as part of our starting definition? They come from stars. Everything in this room, everything on the world, everything in each of our bodies, in fact, every atom began in a star. At least every atom heavier than hydrogen, it turns out. Heavy atoms are made by stellar processes. So this is the astro part of the word astrobiology. When we look at a galaxy, like this whirlpool galaxy, we see literally hundreds of billions of stars. There's about 400 billion in our galaxy, for instance, the Milky Way. And not only do we see stars, but we see these dark clouds. That is stardust, literally stardust, particles of matter produced by dying stars when they go through an explosion that we call a supernova or just ordinary novas. And those dust clouds then gather gravitationally into things that are called globules. And right up there we have the start of a new star. And if you see the magnification, here is an actual image of a newly born star with all the dust around it left over from this molecular cloud up here. And as it begins to glow, you're going to see that planets are produced. So we are stardust. Kind of fun to really know what happens. I'll give you a little nuclear chemistry here. Take this home and impress your friends at a party. You now know where the carbon in your body comes from. This was discovered or proposed by Fred Hoyle, very well-known British astrophysicist back in 1947. Fred realized that when a star reaches enormously high temperatures. This is about 10 times the temperature of our sun, 100 million degrees. You begin to go through a process by which alpha particles, 3 of them, come together to produce carbon. So here's a couple of helium nuclei, which are alpha particles, crashing together at these extremely high temperatures, producing a beryllium nucleus. That's the lightest metal, in fact. And that is followed by another collision with a third helium, or alpha particle, to produce carbon. And if you count up the protons, you'll see there's 6 red protons, 6 black neutrons, and that is, in fact, the ordinary form of carbon that makes up all life on Earth. A little bit before that, people had proposed that if carbon was present under these conditions, that the carbon, in fact, could be hit by a proton, just a single proton, that's a hydrogen nucleus, pop out a helium. Excuse me, here it is. The carbon is hit by the proton, pop out a gamma ray, and become nitrogen. So suddenly we not only have carbon, we also have nitrogen. The nitrogen can pop out a positron and a neutrino, become another form of carbon. This is a slightly heavier isotope called C13. That can be hit by a proton to produce another form of nitrogen, another stable isotope, hit by another proton to get up to oxygen. That can then lose a positron and become another nitrogen, which pops out of helium, and this thing goes around and around in a circle, spewing out oxygen, nitrogen, and carbon as part of that cycle. This is the stuff that accumulates in stars, and this is really one of the fundamental discoveries of astrophysics in the last century. Fred Hoyle should have gotten the Nobel Prize for this, but there was a little problem with Fred, and that is that not only did he write good science, he wrote good science fiction. So really beware of this if you're interested in a ticket to Stockholm sometime. It's very clear that a few people that indulge in playful science, like Hoyle, like Gamow, that they do not make it to Sweden. I see a hand back there.
Could I ask a very brief question? Was that ever experimentally verified?
Sorry again?
Was the cycle ever experimentally verified?
Oh yeah. We can see the spectra of these nuclei and in the light that is being produced by sun. So it's really very well worked out. There's a lot of math, physical math that goes into this as well. So it's really the accepted way for this to work. It has to be at very high temperatures. We couldn't do this on the Earth. We can make hydrogen bombs, but we cannot make this sort of thing because it's too high a temperature. Well, that stuff would get stuck in the star, except that stars have a lifetime. When stars burn up most of their hydrogen, they undergo a collapse. And during the collapse, they spew out a massive amount of the stuff shown in these 2 planetary nebulae. So these 2 stars, you can see them in the center, have reached the end of their lives. They have lost a lot of their mass to the space around them. And by the way, these things would be maybe a light year or so in diameter. These are big objects, even though the star looks big, it's just really a point of light there. And of course the astronomers, this is the only time that astronomers can exercise their imagination. So whoever sees this first, it's like the cosmic Rorschach test. This is the Eskimo Nebula, and this is the Cat's Eye Nebula, and that's the names that are stuck. Well, that looks very exotic, all this stuff out here, but this in fact is what we are made of. This is nothing more than dust particles, and this is what one looks like. This is an interplanetary dust particle that is picked up on a high-flying aircraft. They used to use U-2s to collect dust particles from the upper atmosphere. And it's a silicate mineral core. So most of what you see here is a quartz-like or sand-like or glass-like material. And then on the surface, at least when it is out in outer space, is a mantle of ice. So these things run around out there in those molecular clouds collecting gas like water vapor, like carbon dioxide, methanol is out there, methane. All of these things collect on these particles. So when I showed you those molecular clouds, that's why they're called molecular clouds. They are dust particles like this one with a surface coating of molecules. And that's where new solar systems come from. Let me show you how that works. Here's a beautiful picture of a newly formed group of stars. They're very blue because they're rather new. Their star light, the pressure of the light, has been forcing all this dust away. So they've cleared out the space around those stars. One of them has already exploded. This one has gone through reached the end of its life, and you can see that ring of matter around it. If we saw that from the top, you'd recognize a planetary nebula, such as I see. And this is a puff of stuff coming out from either pole. When the star collapses, it puffs out material along that axis there. None of these stars is going to make planets and solar systems. The reason is that their starlight has cleared the dust away. But look down here, right there. There is a dusty star. That is what a new solar system looks like before it has gone through the process of planet formation. Now I'm going to show you a little movie. I hope. Come on. There we go. So this is a molecular cloud, and what you're going to watch in just a few seconds' time is a collapse of that cloud into a spinning disk we call a solar nebula. In the center of the disk, you'll see a new star formed, and in the leftover material, you'll see new planets. So watch what happens here. Notice the rotation. This is just turbulence that turns into rotating matter. There's the disk forming. It's also rotating. That's why planets rotate around the sun. It's the leftover momentum. Here's the star collecting material gravitationally from the disk. The star begins to start up its hydrogen fusion. Lots of light is produced, and heat and other energies like those solar winds that we heard about. And here, left over, is the planets circling that star. This happens rather fast. It's just 100 million years or so. You can go from start to finish, at least in these computer simulations. Planet formation is a violent affair. It's not just a gentle sifting of dust and gas down to a newly born planet, but in fact, it looks more like this. This is the Earth, and it was only a few hundred kilometers in diameter, about the size of a big asteroid. And you can see these collisions between smaller objects, and the larger object is attracting them gravitationally. Heat is produced. That heat is captured in the interior of the Earth, and the heat that drives Mount Fuji and the other volcanic areas of the Earth is leftover heat trapped in the Earth by its original accretion process. The Earth was unique in our solar system by undergoing a collision with an object nearly the size of the Earth. It's now the consensus view that the Earth-Moon system was produced about 4.4 billion years ago when a Mars-sized object happened to collide with an Earth when it had about maybe 90% of its present mass. The Moon then congealed, accreted out of a ring of material. We're now standing on the surface of the early Earth. This is the ring out of which the Moon is accreting. The Moon was much closer at this time, so it really did nearly fill the sky, as you see here. The most important point for the original life is that these temperatures, red hot temperatures, are what we call pyrolytic. That means that organic material cannot hang together. It all turns into gases like carbon dioxide and water vapor. So life could not begin until the Earth had cooled down and oceans had formed. So this is maybe 100 million years later. Things have cooled down pretty well. The Moon is still being cratered by impacts of asteroid-sized objects, and we still see those craters on the Moon today. The Earth is covered with a film of water, just a few miles thick, and that, of course, is the water that's going to give rise to life. But I just told you that all of those organic compounds that might have been available on the Earth before the moon forming event had been completely damaged, completely turned into gas. So how did we get organic compounds back? Several ways, but one way that we agree upon is that they were delivered to the Earth by the impact of comets and meteorites, and the artist has shown some of these little impacts here. So next time you look at the ocean, just think to yourself that that ocean was delivered to the Earth after the moon forming event. And along with those comets, and along with the dust that was sifting down, and along with the asteroids that were hitting the Earth, came organic materials. That organic material is produced on the original dust, as I've shown here. This is one of those interplanetary dust particles I talked about. And there you see that layer of ice on the surface. And that ice, in the presence of ultraviolet light, and with these simple compounds frozen into the ice, water, methanol, carbon monoxide, and ammonia, the UV light gives energy, chemical energy, that produces things like amino acids, ketones, what else? Quinones, amides, amphiphiles. Keep that word in mind because that's going to be very important for the rest of what I say. This stuff then accretes to form the comets that go by the Earth once in a while. The interplanetary dust particles that sift down to the Earth. Even today, 30,000 tons of material in the form of dust gets to the Earth every year. It's really quite astonishing. We can measure that. And of course, things like planetesimals. These are asteroid-sized objects that come together to form planets. They bring their organic compounds along to the Earth's surface, and that's where at least some of the organic material came from that gave rise to life. Well, how do we know? The reason we know is it's still happening today. Every time you see a shooting star, or once in a while a big bolide like this one here that just by pure luck was captured on film, then these things come in. They are delivering material to the Earth. We're in the last long drawn-out phase of the accretion of the Earth that started 4.5 billion years ago. Now, this is like a meteorite that fell in Australia about a little over 30 years ago, 1969. And that meteorite is called the Murchison meteorite, and that's something that we are studying in my lab. These are called carbonaceous chondrites, or carbonaceous meteorites, because they have organic material in them. Those same organics that give rise to life are being delivered even today as meteorites containing organic compounds. So this is about a golf ball-sized object. This is the fusion crust, because these things come in pretty fast, of course. The surface actually melts, but the interior stays very cold. And what we do is to break that open. I'll just show you one of these that we were breaking open. Very easy to break open. It's kind of like sandstone, so you just take a chisel, and you try to do this as under clean conditions, of course. You crack it open, and when you look at the inside of the meteorite, this is a freshly fractured surface, you see these beautiful glowing objects. These are the size of cells to big cells, and that's the organic material of the meteorite. 2% of the mass of this stony meteorite is made of organics. Now, when we extract these organics with a solvent system like chloroform, for example, and we allow them to interact with water, this is what we found. It's about 15 years ago, we reported that the organic compounds called amphiphiles that are present in carbonaceous meteorites are able to form cell-sized membranous structures. We know they're membranes because they can capture fluorescent dye molecules, as you see here. They're like little bubbles floating around in the microscope. So we proposed at the time, and I think it's just a general consensus now, that this is the way that the compartment that made up early cells was being made available on the early Earth by organic compounds delivered from the extraterrestrial infall to the Earth's surface. Well, how did that turn into cells? And that's the next part of my story. Let's just think about what cells are. Self-assembly is one of the primary things that occurs in life today and must have been part of the process of the origin of life. boundary structures, what I'm saying there is just the outer layer that forms the compartment within which the rest of the cell lives. So that's self-assembly. The other thing that makes life possible is what we call directed assembly. That's when a gene directs the assembly of another polymer, of it like a protein. So when you have catalyzed growth and replication of these polymers, then you have what we would call a living cell. So that's what we want to do. First, we want to self-assemble structures that have the potential to do this. Then we want to find ways for directed assembly of polymers to begin. Then we would know what life is, at least chemically. Let me just tell you a bit about self-assembly. When single strands of DNA assemble to form a double helix, that's called self-assembly. When a protein is produced by a ribosome, it folds up into a very precise structure. That's called self-assembly. And when lipid dispersions self-assemble into membranous structures, that is called self-assembly. The molecules that we find in the meteorite are called fatty acids. Here's a fatty acid. It's got an oily part of it that loves to be dissolved in organic supplements. And it's got a water-loving part, hydrophilic part. This is an acid, it turns out. You can see it's an acid because it can lose a hydrogen ion. That's what acids, by definition, are in chemistry. So these molecules are able to self-assemble, and every one of you has done this in your lifetime. Whenever you blow a soap bubble, that is a self-assembled membranous structure. It's as simple as that. So what we do in the laboratory is to make self-assembled microscopic bubbles. And if we could look at the molecular level at the self-assembled membrane, what we would see is hydrocarbon chains all in the interior. There's actually a double layer. This is one layer, this is the second layer. And that oily part is what separates the outside of the cell from the inside. Here are the head groups of the lipid, more head groups over here, and everything you see here is water molecules. Now you know that oil and water don't mix. And if you look here, you might just see one water molecule right there. And that's about right. One molecule per several hundred chains is how much water actually would be able to get across the bilayer by diffusion. It's a pretty good barrier to the movement of molecules like water and ions and glucose and all these things that cells like to do and use. So here's some membranes we've made in the lab from a very simple mixture of a fatty acid. This is a 10-carbon fatty acid, just as I showed you, mixed in with a 10-carbon alcohol. You don't have to have that there, but it helps the stability. They've been stained with a fluorescent dye, and these are about the size of living cells. So it's as simple as that to make the boundary of cells. Then, if we add a solute, like a macromolecule, for instance, DNA would be one such solute, or a protein, and in this little drawing, I show those as these lines, these rod-like structures. Here are the membranous vesicles. If we put it through a single drying process, all these vesicles fuse to make these multilamellar structures. You can actually see these under the microscope. Then when we add water back, we discover that some of the molecules have been captured on the interior. These molecules could not get in all by themselves, but if you put them through this drying process, they do. Now, I'm going to show you that when you do this in real life, under what we call simulated prebiotic conditions, we can make some of the molecules that are now important to life, in particular RNA. So keep this little process in mind, this drying and wetting process. That's an important point. Here's some DNA captured in lipid. Makes these beautiful structures under the microscope that we can see by adding a fluorescent dye. After a while, the structures break up and turn into vesicles. And these vesicles then are filled with fluorescent DNA that's been stained by a fluorescent dye. We're just seeing these under the microscope. A human red cell would be about this size. It's about maybe 10 microns across. This might be a liver cell, and the smallest ones you see would be the size of bacterial cells. Just to give you an idea of the size of these. So it really is easy to make what we call protocells. Just mix in a lipid, you put in some of the material you want to encapsulate, go through a single drying wetting process, and you come back and you have this cinder stuff. So the question is, could that occur on the primitive Earth? So we're asking that question now, and that took us to Kamchatka, Russia. So we're going to find out whether we can actually make this process I described occur under simulated models. Well, not even simulated. These are real models of what we think the early Earth was like. So I'm going to give you my perspective on what the Earth was like about 4 billion years ago. Here's an artist's version. The early Earth was volcanic. It was much hotter than today. In fact, you may have heard one of the speakers earlier say that archaea, these thermophilic bacteria, are some of the most primitive organisms in terms of their gene line that we can trace. So we think that life probably began in a rather warm environment. The temperature here was probably 60 to 70 degrees Celsius. Of course, we have boiling hot springs associated with the activities the artist shows here. And we have volcanoes spewing carbon dioxide and other gases into the atmosphere. There's no oxygen. Life did not advance to the point where they could even produce oxygen as plants do for us now. It's a carbon dioxide atmosphere mixed in with nitrogen. So that is the idea about where life began. We're going to go fly off, and I've been here twice now, just a couple years ago, in fact, most recently, to Kamchatka. Now, Kamchatka is important to us because it's so far away from every place else on the Earth. It's a high altitude. The mountains, the volcanoes there are up to 10,000 feet. High latitude, that means that plants don't grow very well when you get up onto those volcanoes. And furthermore, it's very volcanic. So there are sterile sites that are just totally free of other life. So it's like the early Earth in the sense that it's sterile, there's nothing alive there. We can do experiments that we can't do elsewhere. So this is what Kamchatka looks like from the air. Japan, by the way, is right down here. Japan almost touches some of these little islands in between. And Japan, Mount Fuji, is part of this range of volcanoes that goes up the point at which the Pacific plate slides under this part of the world. So that's these are all the volcanoes. Petropavlovsk is right in here, so when we fly over from Anchorage, we land there. Then we take a Land Rover type vehicle to Mount Mutnowski, which is that volcano right there. And the reason we use that is that we can get to it. These others all require helicopters at $2,000 a day to get there by helicopters. That's a little expensive for our taste. So here we are now climbing. This is in the summertime, late summer. There's quite a bit of snow remaining still. And we're climbing at about 6,000 feet now, just getting over the lip of Mount Mutnowski. We're going to look down into the active crater that last erupted about in 2000. Here we are looking down into what I think is a good model for the prebiotic Earth. No life, there's no plant life, maybe a little microbial life if you really look. This is a fairly sterile environment. So we would then work our way down into the crater itself, and in the crater you find places like this. And that's me collecting some material from one of these sulfur hot springs. We have to wear a gas mask, by the way. There's a lot of sulfur, hydrogen fluoride in the air. And I'm taking a sample from a boiling mud pit here. I'm going to pull it out, and the next one shows that sample. So we would take this, we would filter out all the sort of ash-like material, and then take the water home to analyze in a laboratory. So we're looking around then for a place that looks like where life might begin. Now, we didn't find any organic material here. It's perfectly sterile, nothing that we could see at all. So we wanted to find something a little more conducive to doing experiments, and we found this little hot spring. It's isolated, as you'll see. It just boils in the center there, and it's filled by a little bit of snow melt that comes in from beside the spring. And this, I think, is an example that I would use as a model for where life might begin. And the reason is that on the edges of the hot spring, you have those wet-dry cycles that I just told you about. So here's a place where, as it dries, material is concentrated. If there's lipid-like material there, it'll form membranes around anything that's there. It's a place where a lot of natural experiments can go on. The sort of experiments that I think would lead to the origin of life. So we did some work in the laboratory. We went back to the laboratory with these ideas. And now I'm going to show you how we could use that model system, simulating it in the lab, to make RNA. Oh, yeah, let me say RNA world. You've heard this before. This is how life was. We don't think that life sprang up as our kind of life. We think that life went through what we call an RNA world, where RNA was both the catalyst and the gene carrier. And the reason for that is that we don't have any better ideas, to tell you the truth. It's just a way that we can get at this experimentally. So what I'm asking is, how could the first RNA be synthesized? And what we have discovered is that we have a source of mononucleotides, and these are the monomers that string together to produce a piece of RNA. We can drive polymerization reactions using those fluctuating wet-dry cycles, such as I showed you in that volcanic region. And here's the basic concept. If you're a chemist, you'll follow this easily. If you mix RNA or any other polymer of life with water, it will turn into its monomers. It's a process called hydrolysis. Water is added to the bonds that hold it together, and you end up with the 4 monomers of RNA. These are called nucleotides. This is called adenosine monophosphate, uridine monophosphate, guanine monophosphate, and cytidine monophosphate. So those are the 4 that will make RNA if we give it a chance. Now we can pull this reaction in the other direction by removing water. So if we can find a way to get water out of it, this reaction will run in the other direction toward RNA. So that's all that we're going to try to do. Here's what AMP looks like. So you're getting a little chemistry lesson here. This is adenine. That's the A of DNA and RNA, adenosine monophosphate. There's a little sugar group, and there's the phosphates. That's all there is to it. Pretty simple. So we're assuming that things like this were around on the early Earth. Doesn't have to be this exactly. And what we're going to do is to put this through those wet-dry cycles that I told you about, but we are going to have lipid present, and that's the trick. We found that in the absence of lipid, nothing happens when we dry it down. But in the presence of lipid, something rather magical happens. We're just going to dry it down, rehydrate. We're going to do that up to 7 times. We're going to have some lipid there, and a little bit of elevated temperature to mimic what we would have in a volcanic region, and we're going to measure polymer synthesis as RNA. So this is what we think happens. We think that the AMP is trapped between the lipid layers. Here's a lipid bilayer, see it's trapped. And when it's trapped, it's organized because it doesn't have anything else to do. No more degrees of freedom. So it probably looks something like this when it gets trapped between the lipid layers. And all we're going to do then is to warm it up a little bit so that water has a chance to leave. There's the water molecule, and if it leaves, you can make RNA. So that's all we're trying to do with this. And it works. This paper is in press. It'll appear in the next week or so, in fact. The only one that you have to look at is this one over here. This is radioactive RNA. What we've done is to treat our product from that reaction I just told you with a couple of enzymes that are known to act on RNA and add a radioactive group on the end of the RNA. That radioactive group exposes film. And so if radioactivity is there, we'll see a shadow on this film. So here's 1 cycle, 3, 5, 7, and you can see the RNA building up as we put it through cycle after cycle. You can also see this is 20 bases. This is a standard from 20 to 100 known RNA fragments, and it gets labeled as well. So there's 20 bases, 30, 40, 50, all the way up to 100. So you can see that we're making long strands of RNA by this process. Everything else here is just a variety of experiments that test the idea. The best part of all is that when we end up, we don't end up with just a clear solution of RNA, we end up with vesicles like this, lipid vesicles, about cell size, and there's the RNA stained with fluorescent dye. You can see it accumulating in these vesicles. So if I had to guess what the first forms of life look like, it'd be something like this. Lipid vesicles containing a genetic material that also had catalytic activity, and that's what we're going to try to do. Well, I will finish up now. We're going to try to produce artificial life in the laboratory. That's kind of like playing Frankenstein, isn't it? Except we're doing it with the microscopic level. And as I say, this work is going on here in Japan. Some of the pioneers are here in Japan. It started about a little over 10 years ago when two laboratories, Luigi Luisi's lab, he was in Switzerland then, and our lab, we both showed in about the same year independently that you can make RNA using a molecule called ADP, which is just like AMP, adenosine monophosphate, that's got an extra phosphate on it, and that an enzyme captured with the ADP will produce RNA in the vesicle. So that's where it started. Next thing we did was to show transcription. And this paper just was published about 6 months ago, so you're right up to date on this. We encapsulated a polymerase. We used a DNA template. We had NTP substrates added to the outside of these model cells, and we found that we could transcribe the base sequence from DNA into RNA. You can study the papers and philosophical transactions B a little earlier this year. This is what that looks like. There's the enzyme. Here's the template that is being used. It's a phage DNA template. Here's the RNA that's being synthesized. We have found a way to make transient defects in the membrane just by heating cycles, and that allows all these substrates to go in to supply the enzyme. And this is what it looks like when you get done. We can actually see the RNA accumulating in these vesicles by staining it with a fluorescent dye. And once again, this to me would be what I would think a primitive form of life would look like if we could see it nearly 4 billion years ago. Okay, now here's some Japanese work. This is really very interesting stuff. They're making proteins. And this work, the pioneer was Yu et al. Working in Yomo's lab with Kaneko's being a colleague on that. Another paper from Nomura et al. In 2003. And what they're doing is putting ribosomes into it. They're getting these out of E. coli. This is standard bacterial system. They're popping the E. coli and taking everything that's in the E. coli and trapping it in lipid vesicles. And then they're seeing whether they can make protein. And the protein that they're making is a fluorescent protein called green fluorescent protein. And that's just a marker to show that it's actually working. So here's what their vesicles look like. There's the ribosomes. There's the messenger RNA for the green fluorescent protein. And there's the protein being produced by the ribosomes. They wait around a little while, and the vesicles contain green fluorescent protein. This is work that came out 3 years ago from Rockefeller, Moreau, and Liebschaber, and they found a way to put a hole in the membrane. And that's important because you really want to be able to feed your cells. So not only did they make green fluorescent protein, but they made a channel-forming agent called hemolysin, HL, that punches a hole in the membrane, and now stuff on the outside to get to the inside and feed that. The last one that I'll tell you about is an encapsulated genetic cascade. This is by Ishikawa et al., 2004. And what they did was to put in a piece of DNA that had a gene both for a polymerase and for green fluorescent protein. They were then able to use what is called flow cytometry to see whether it worked, and it did work. I won't try to explain this, I don't have time, except to say that in the complete system, all of these little dots are single vesicles passing through a laser beam, giving flashes of green light to the green fluorescent protein that was made. And here are a couple of controls showing that it doesn't work unless you have a complete system. So we're getting very close to making artificial life. And I'll just finish up. This, I think, is what we're aiming at, that it can be done. Nobody knows that it can be done, but this, in my opinion, would be able to be claimed to be synthetic life. Now, we're cheating a little bit. We're starting with ribosomes, because we don't know how to make ribosomes from scratch. Everything else we could, in a sense, make if we wanted to. So here's a big bit of DNA. It's got about 100 genes in it, most of which are just used to make the ribosomes in the translation system. You've got one to make the pores that are going to let nutrients in, and one to make lipid. And when you let that run, it might work. But there's a remaining question. This is my last slide. How do we really do it? We can make an initial synthesis of catalytic and replicating polymers. And that's what I showed you that we can do now. We can make an RNA that could be both a replicating polymer and a catalytic polymer that perhaps could lead to the RNA world. Then we have to find a way to make ribosomes. This is a big stumbling block. Nobody knows how to make ribosomes yet. And yet this is the source of life today. Without ribosomes, you're nowhere. We need to find a way to reproduce those systems. And they reproduce themselves. It's sort of the autocatalytic cycle that they go through. We have to have a source of energy and nutrients. We need membrane growth, because you can't have stuff growing inside unless you have the membrane growing as well. And you need a regulatory feedback between the membrane growth and the stuff inside. Otherwise, one of them will outgrow the other, and you end up with a bag of nothing. And you have to find a way to divide. I think we can be optimistic. Life did it all by itself. Nearly 4 billion years ago, all that I told you happened through just simple self-assembly and evolutionary selection. So that's my story. This is supported by NASA. And nice support from NASA. These are some of the people that were involved in the work. I thank you for your attention.
Thank you very much. Very nice and exciting talks. We have a time to asking a comment. Okay.
Thanks for a very good talk. I have a couple of questions. I remember years ago there was a lot of attention to electrical discharges, lightning in this early forms of life. Do you think that those kind of studies survived, or?
Yeah, it's a good question. Stanley Miller, 1953, used a spark discharge. And he sparked a reducing gas mixture, hydrogen, ammonia, methane, and water vapor. And he got amino acids. And this really broke the field wide open. So it's a landmark discovery of Stanley's. By the way, he died just a few months ago. And he's sort of left his contribution for us. So that's why electrical discharge was used. It was convenient to use. Since that time, we've shown that ultraviolet light can do the same thing. Certain temperature ranges can do the same thing. There's a variety of ways to make complex molecules out of simple starting mixtures. Lightning is a minor component of the energy available in the early Earth. The primary energy that would have been available is ultraviolet light from the sun, as well as volcanic heating. Those are far outweigh electrical discharge as a way to drive these syntheses.
And I have a small technical question. I missed the connection between what you did on Kamchatka with this last part of the talk. So you just wanted to establish that it's very sterile?
Well, yeah, there's the Kamchatka stuff is a whole other talk. What we did was to pour in a mixture of the sorts of things that I actually did in a test tube that I showed you. I just didn't have time to go into all of that. And what we found was interesting, disappointing in a way, but it certainly guided us, because everything we dumped in got stuck on the clay surfaces and was immobilized. You know, that pool is filled with clay. So this is something that we have to learn how to get around, is how do you keep everything that is being made, organic materials, from sticking to mineral surfaces and being removed from reactive diffusion. I think that maybe the way to do that is, instead of going to Kamchatka I'm going to have to go to Hawaii. I just can't stand the idea. But Hawaii does not have clays. Their volcanic activity goes right on to lavas. Clay is sort of characteristic of continental land masses like Kamchatka, whereas a Hawaiian surface is almost like glass because it's a glass-like material. So there I think we might have more luck with that experiment, and that's on our to-do list. Ready?
You mentioned clay just now, and I remember there was a man called Cairns who was interested.
You mentioned clay, and there was a scientist called Cairns who was interested in the idea that crystalline clay structures could have been precursors for life. Does that play into your plans?
Yeah, it's a great idea. Graham Cairns Smith, actually. He's at Glasgow University. He's a mineralogist, geologist. And he came up with this idea, and it's a nice idea. Got a nice Scientific American article out of it, and he wrote a book on this, that clay, in a sense, is a primitive organizing surface, and that if you can stick molecules like nucleotides to the clay, you might be able to line them up in a way that's conducive to their polymerization. So in a sense, what I showed you with the lipids is along the line of that idea, but instead of a mineral surface, we're using a lipid surface to organize the stuff. Now the problem with clays is that everything that you make sticks so tightly that it's hard to get back off. Furthermore, you have to totally saturate the clay surface with what you want to make. When we did that with that Kamchatka work, we dumped in gram quantities of amino acids, of nucleobases and phosphate and so forth. And what it comes down to is that when you do the calculation, there was so much clay in that little basin that it was a surface area the size of this room. It's about 100 square meters per gram of clay, the surface area. So there's one molecule here, another molecule here, another molecule here, and they just can't get together to react. So they became immobilized as potential reactants. So that's the problem with clay. Yes, it does absorb the stuff, it concentrates it, but it also inhibits their diffusion that's required for reactivity.
It's an amazing talk, Dave. I've never heard that story as you told it. I didn't hear that story at all. But one thing which emerges is that it seems to be all too easy to create life. And in that case, isn't it an extraordinary coincidence that it only seems to have happened once on Earth? At least if you look at the phylogenetic tree, it seems to go back to a single origin. Shouldn't there have been lots of origins? And maybe, do you think there were, and that they all got eaten by one particularly successful branch?
Yeah, it's a lovely question. A lot of people thought deeply about that. The early Earth was being bombarded by vast, 100-mile diameter asteroids at that time. We see the remains of that on the moon in the form of the cratering record. Any one of those really large ones would have totally vaporized the ocean, and anything alive would have been killed. So we think that the most plausible ideas of life, yes, it does start easily, but it was extinguished except for the deepest thermophilic life, and that is a bottleneck through which life passed. So not so much that it's just one origin of life. It started up easily, but there's only one bottleneck through which life today passed, and it was produced by this bombardment 3.8 billion years ago. So, yeah.
What do you think life will be? Because irrespective of being or becoming, I do believe the principle of life itself must be the same. So from the past to the future, I do believe the same universal principle might be present. And to such universality or universal principle can be scientific work.
Okay, so I just to summarize your questions, I understand and make sure I got it right. First off, what will life be? That's one I can tell you just a little bit about. But as soon as I get outside of the sort of synthetic life that we have control over, nobody predicts what evolution can produce in the way of life. We see evolution all in hindsight. And Stephen Jay Gould has commented that if we could run the tape of life once again, we'd probably end up with something entirely different, which suggests that it's not predictable, at least at the level that we exist now. There's no guarantee that intelligence would have arisen on our particular form of life. So what I think life will be, though, is that we will get control of bacterial-like cells. People like Craig Venter are actively now trying to engineer bacteria to do exactly what they want them to. We talk about synthetic biology, where we have a toolkit of genetic tools that we can use to literally, like engineers, make the bacteria do this and that, and come out with some desired pharmaceutical agent or a protein that we need, just as Genentech did with insulin so many years ago. So I think that's what life will be, at least at the synthetic single-cell level. At the higher levels, I think it's anybody's guess because of the vagaries of evolution.
Do you believe there is some universal principle independent of the level of evolution?
A universal what?
Principle.
Oh, universal principle. Ah, yeah, okay. Well, yeah, I believe in the laws of chemistry and physics. And I think that what I just told you here could happen on Mars, and maybe did. There's an amusing point that people love to make, if you haven't heard it before. We might be Martians. If life began on Mars first, it could very well have been delivered to the Earth. And when and if we do find evidence for either extinct or existing life on Mars, which could be microbial in some of these deep hydrothermal areas, we might discover that it has the same genetic code, the same DNL amino acid distribution and so forth, and that we in fact either we're Martians or Mars-like is terrestrial, which is less likely. So there is a universal organizing principle. It's called the laws of chemistry and physics, and I think life could arise anywhere, but it would not necessarily look like what we have on the Earth.
Other question? Why?
Very nice thing today. And make one small correction. First of all, it's very nice of you to have been fed for that. Right?
Just a little bit louder, please.
About Fred Hoyle.
Oh,
yes. Well, I don't think really is anything to worry about. It's don't think to do with the science fiction. He was, Fred Hoyle was the orthodoxy. He was the theory of continuous creation. Higgs force out the Young Turks out of the department as the people who wanted to bring in the Big Bang theory. And so he was sidelined, and he got totally fed up with everybody, and then he just walked out one day. And nothing to do, I don't think, with his novel, I think.
Yeah. Well, yeah, Gamow is the other example I used. And people pretty well agree that the reason Gamow, who proposed the Big Bang, the original Big Bang, turned out to be correct, the reason he was not recognized with the Nobel Prize, perhaps he didn't live long enough, but it was because he wrote children's books. And people are on record saying that sort of thing. Fred Hoyle, besides his good science, was in fact a science fiction writer with maybe half a dozen books. I read some of them. He also had some ideas that are way out there in a very real sense. He claimed that the Archaeopteryx fossil in the British Museum was a fake. He also believes that, and still does, he has a colleague, Vikramasinghe at Cardiff, who thinks that in fact outer space is filled with spores of life, and a firm belief in panspermia, that life on the Earth did not develop on the Earth, but was brought to the Earth in the form of panspermia-like spores. So these are far-out ideas, and yet he published them, he wrote books about them, and I think that may have damaged his reputation. But thank you for the historical comment on somebody who was there when a lot of this happened. We could chat a little bit later about that.
One last question.
Okay.
Okay.
I am a theoretical thesis studying the...
Okay, this is better.
I'm a theoretical thesis studying the structure of the whole state. I want to ask definition of life. Our life is based on the fact that the nucleus oxygen-16 is a very stable nucleus, and also the structure, atomic structure of oxygen, electronic structure is very active. This is very important. So, our life is composed of carbon and nitrogen, oxygen, CNO element. And so this is our life is a baryonic system composed of variant of light nuclear element. So if you define life as a self-assembling system is a boundary structure, do you expect life which is composed of heavier elements like calcium-40?
Life is composed of what?
Heavier
Heavier.
elements like calcium-40. So if you have some structure, we could define such kind of baryonic system.
Okay, I think that I understand your question. It so happens that life uses carbon, oxygen, nitrogen because they're very common, because they have certain special properties. I think your question is whether heavier elements could also have been used, or maybe we could use them to produce artificial life. Is that your idea?
Yes, yes.
Right.
Yes.
Well, you know, we have these science fiction writers like Michael Crichton, who proposes nanotechnology run amok and taking over parts of the world. So I think this is still at a science fiction level. It's just amazing how these little molecular machines produced by carbon, oxygen, nitrogen work. We have true machines in our bodies working at the molecular level. I don't think anybody yet knows how we could do anything like that by compounds other than carbon, oxygen, nitrogen compounds. It's just difficult to imagine. It might just be the limits of our imagination, of course, but my answer to you is that I sure don't see a way to make life from heavier elements in the absence of carbon, oxygen, nitrogen.
Thank you. Because of the time, I just want to stop here. Thank you very much again.